Learning goals. Welcome to this learning session on photosynthesis, plant energy and food production. In this course, we shall address photosynthesis, introduction and equation, mechanism, components and energy transformation, key limiting factors affecting photosynthesis, investigating factors through experiments, photosynthesis and plant respiration. There will be a quiz at the end to test your knowledge.
Introduction to photosynthesis. Photosynthesis is the essential biological process by which plants and algae convert light energy into chemical energy, producing their own food. This process is fundamental to life on Earth, forming the base of most food chains. Organisms capable of photosynthesis are known as producers because they generate organic compounds from inorganic sources. They are the primary source of food and biomass, which is the total mass of living material at a specific stage within a food chain. For example, phytoplankton, microscopic algae in oceans, are responsible for producing roughly half of the world's oxygen through photosynthesis. The photosynthesis equation. The overall process of photosynthesis can be summarized by a chemical equation that shows the reactants and products involved. Carbon dioxide and water are the raw materials, which are transformed into glucose and oxygen. This reaction requires light energy, which is absorbed by chlorophyll, a green pigment found in plant cells. The balanced chemical equation for photosynthesis is 6CO2 plus 6H2O, CH plus 6O2, illustrating the precise molecular ratios involved. Role of chlorophyll and chloroplasts. Chlorophyll is the green substance found within the chloroplasts of plant cells, primarily in the leaves. This pigment is crucial because it is responsible for absorbing the light energy needed to power photosynthesis. Chlorophyll absorbs light most strongly in the blue and red portions of the electromagnetic spectrum, while reflecting green light, which is why plants appear green to our eyes. There are several types of chlorophyll, with chlorophyll A and chlorophyll B being the most common in plants, each absorbing slightly different wavelengths to maximize light capture. Energy transformation in photosynthesis. During photosynthesis, a crucial energy transfer occurs. Light energy is converted into chemical energy. This chemical energy is then stored within the bonds of the glucose molecules produced. This conversion is a complex process involving two main stages, the light-dependent reactions and the light-independent reactions, Calvin cycle. The light-dependent reactions capture light energy and convert it into ATP and NADPH, which are then used in the Calvin cycle to fix carbon dioxide into glucose. This intricate energy transformation ensures that plants can sustain themselves and, in turn, support nearly all other life forms on Earth. The rate of photosynthesis is influenced by several environmental factors, with light intensity being one of the most critical. As light intensity increases, the rate of photosynthesis generally increases because more energy is available for the light-dependent reactions. However, this increase is not indefinite. Eventually, other factors like carbon dioxide concentration or temperature will become limiting. For instance, in a dense forest, plants on the forest floor often have broader leaves to maximize the capture of the limited light that penetrates the canopy, demonstrating adaptation to low light intensity. Experiment, light intensity and CO2 setup. To observe the effect of light intensity on photosynthesis, an experiment can be set up using algae and a hydrogen carbonate indicator solution. A student prepared 6 bottles, each containing the same number of gel beads with algae and an equal volume of the indicator solution. The hydrogen carbonate indicator solution is a pH indicator that changes color based on the carbon dioxide concentration in the water. It turns yellow when carbon dioxide concentration is high, orange-red at normal air concentration, and purple when carbon dioxide concentration is low. This allows for a visual assessment of photosynthetic activity. Experiment, light intensity and CO2 results. After 2 hours, the bottles in the experiment displayed distinct color changes, reflecting different rates of photosynthesis. Bottle A, placed closest to the lamp, turned purple, indicating a very low carbon dioxide concentration. In contrast, bottle B, positioned furthest from the lamp, turned yellow, signifying a high carbon dioxide concentration. The bottles in between showed a gradient of colors, from orange-red to lighter purple, demonstrating the direct relationship between light intensity and photosynthetic activity. Light intensity, CO2, and respiration. The observed color differences in the experiment are explained by the interplay of photosynthesis and respiration. In bottle A, high light intensity allowed photosynthesis to occur rapidly, consuming carbon dioxide at a much faster rate than respiration produced it, leading to a very low CO2 concentration and a purple indicator. Conversely, bottle B received very low light, meaning photosynthesis was slow or negligible. However, the algae continued to respire, releasing carbon dioxide into the solution. This accumulation of CO2 resulted in a high concentration and a yellow indicator. The relationship between light intensity and distance is inverse square, meaning light intensity is proportional to 1/100, where d is the distance from the light source. Limiting factors. Carbon dioxide concentration. Beyond light intensity, carbon dioxide concentration is another crucial limiting factor for photosynthesis. If there isn't enough CO2 available, even with abundant light and optimal temperature, the rate of photosynthesis will be restricted. This is because carbon dioxide is a direct reactant in the Calvin cycle, where it is fixed to produce glucose. Commercial greenhouses often enrich the air with CO2 to boost crop yields, demonstrating how increasing this limiting factor can significantly enhance plant growth and productivity. Limiting factors. Temperature. Temperature also plays a vital role in regulating the rate of photosynthesis. Photosynthesis involves numerous enzyme-catalyzed reactions, and enzymes are highly sensitive to temperature changes. As temperature increases, the rate of enzyme activity generally rises, leading to a faster rate of photosynthesis up to an optimal point. Beyond this optimum, high temperatures can denature enzymes, causing the rate of photosynthesis to sharply decline. For example, most plants have an optimal temperature range for photosynthesis between 20 degrees Celsius and 30 degrees Celsius, with significant reductions outside this range. Photosynthesis as an endothermic reaction. Photosynthesis is classified as an endothermic reaction, meaning it absorbs energy from its surroundings. Specifically, it takes in light energy from the sun to drive the chemical reactions that produce glucose. This absorption of energy is a defining characteristic of endothermic processes, contrasting with exothermic reactions that release energy. The energy stored in the glucose molecules during photosynthesis is a direct result of this energy input, making it available for the plant's growth and metabolic activities, and subsequently for organisms that consume the plant. Plants respire continuously. While photosynthesis occurs only in the presence of light, plants respire continuously, both day and night. Respiration is the process by which plants break down glucose to release stored chemical energy for their metabolic needs. This means that even when a plant is actively photosynthesizing during the day, it is also respiring. At night, when there is no light for photosynthesis, respiration becomes the sole process for energy production. The balance between photosynthesis and respiration determines a plant's overall growth and biomass accumulation, as respiration consumes some of the glucose produced by photosynthesis.
Summary. Well done, you have completed the course, Photosynthesis, Plant Energy and Food Production. You should now be familiar with photosynthesis, introduction and equation, mechanism, components, and energy transformation, key limiting factors affecting photosynthesis, investigating factors through experiments, photosynthesis, and plant respiration. Should you need to revisit any of the topics in this course, please use the menu at the bottom of the screen.